Hello student, today we are going to learn about fermented fish products. As you are aware that fish is a highly nutritious source of protein, and because of the presence of high nutrient content, it is highly perishable. Therefore, fermentation technology is utilized for the preservation of fish, and this practice has been popular since ancient time. The preservation of fish is usually accomplished through a combination of many methods such as smoking, sun drying, salting, fermentation, grilling and frying. These treatments develop a typical flavor and also enhance the shelf life of the product. So, let us know what are the basic principles of fermentation. The basic principle of fermentation is that the microbial enzymes, either of the bacteria or of the fungi, they catalyze the fermentation of the fish. The microorganisms responsible for the fish fermentation are either native, that is, present in the gut, gills and slime of the fish body, or they are externally added as starter cultures. The collective action of native fish microflora present in the salt which is applied to the fish, the body tissue enzymes contribute to the final fermented fish product. The metabolic activities of the microorganism generates certain organic acids like acetic acid, lactic acid in the product. The fermentation process brings about certain changes in the fish, and these changes are either enzymatic or microbiological, which may take place either in the presence or in the absence of the salt. The process results in a complete or partial breakdown of the proteins present in the raw fish. This process results in the formation of simpler stable substances which are amenable for storage at normal temperature. The most important organism during the fermentation of fish are the lactic acid bacteria because of their metabolic capability that converts the carbohydrates present in the fish into lactic acid and other biomolecules as well as energy like ATP. This energy is utilized by the microorganisms for their own growth and development. However, the lactic acid bacteria are of two metabolic types: the homofermentative, which converts 1 mole of glucose into 2 moles of lactic acid, and they also generate a net of 2 ATP molecules. The lactic acid is the major end product formed as a result of fermentation in these organisms. Some examples are lactobacilli, enterococci, lactococci, pediococcus, streptococci, tetragonococci, vagococci, etc. They are capable of fermenting hexose sugars to lactic acid. The second group is heterofermentative. These organisms convert 1 mole of glucose to 1 mole of lactic acid, 1 mole of ethanol and 1 mole of carbon dioxide, and 1 mole of ATP is also generated per mole of glucose. The members In this group include Leuconostoc, Lactobacilli, Oinococci and Vizela species. So, let us see what are the different types of fermented fish products. On the basis of morphology and chemical characteristics, three broad groups of fermented fish products are known. The first is the product is in the form of large chunks, wherein the original form of the fish is retained or preserved as large chunks. Example are Peda same. The second form is the paste form. It is in the form of paste. Example are Nagpi, which is prevalent in Burma. Sauce is the third form, wherein the fermented fish is in the form of a sauce, and during the sauce formation the fermentation is carried out for a longer period of time than in the manufacture of fermented fish as well as fermented fish paste. The sauces are liquid and they contain a mixture of amino acids and other protein degradation products. So, students, let us see what are the factors which affect the fish fermentation process. The first factor is the extent of gutting. The second factor is nature of additives. Third is the chemicals used for fish curing. Fourth are the physical parameters under which the fermentation is undertaken. And lastly, less important are the biological features of the substrate which is undergoing fermentation. Students, as we discussed earlier, there are two modes of fermentation. One is the fermentation which is based on the use of externally added starter culture. Second is the fermentation wherein the native microorganisms present on the fish are active during the fermentation process. So, the starters in the fermentation of fish are either native, which is bringing about the spontaneous fermentation, and these organisms are primarily Bacillus subtilis, Bacillus pumilus, Staphylococcus saprophyticus, Micrococcus luteus and Pseudomonas species. These organisms are present on the fish surface or in the gill or the gut. The microorganisms which are added externally are known as starter cultures. Starter culture is often a mixed culture of molds, yeast and bacteria which is growing on a substrate or a matrix, and usually this is generally a starch based substrate like rice. Starters are also serving as a source of numerous enzymes such as amylolytic enzymes which bring about the degradation of starch into fermentable carbohydrate for the growth of lactic acid bacteria during the subsequent steps of the fermentation process. Example of some of the starters which have been conventionally used are Japanese koji, which is nothing but steamed rice overgrown with Aspergillus oryzae. Some other examples of the starter culture which have been conventionally used are Angkak, which is used in countries like Philippines, Thailand, Cambodia, and the starter culture consists of steamed rice overgrown with a red mold Monoascus purpureus. Longpang is also a starter culture which is used for the production of Thai pla cho, and it is in a physical form of a dry powder, or it can also be made as small flattened cakes. The most common ingredient in all these starter cultures is the rice flour, which is the basal material, and sometimes it is also fortified with other components like vegetables, garlic, ginger, pepper. You must be thinking that the fermented fish products do not undergo spoilage because of a high salt concentration which is present in the product. However, you will be surprised to know that there are certain spoilage organisms which are able to cause the spoilage of the fermented fish, and these spoilage organisms are able to survive at high salt concentration. Some of these spoilage organisms commonly found are the red halophiles, which belongs to the genera of bacterium Halobacterium. These are strict aerobic rod-shaped bacteria and require at least 10 to 15% salt NaCl concentration for their growth. Halococcus, Pediococcus halophilus, Halococcus are also able to survive at high salt concentration of 5 to 10%. These organisms are the two common groups which cause the common defects in the salted fermented fish product, and these defects are popularly known as pink and dun respectively. Some pathogens like typhus bacteria can also survive in saturated salt solution for as long as 3 to 6 months. Salmonella, another pathogen, can survive in 10% salt solution for a period up to 3 months. So, let us see what are the chemical changes that takes place during the fermentation of fish. The first and important change is the degradation of nucleotide and nucleosides in the flesh, leading to the formation of inosine, hypoxanthine, ribose, etc. Reduction of trimethylamine oxide, which is abbreviated as TMAO, a non-volatile and non-odoriferous compound, to volatile trimethylamine, abbreviated as TMA, imparts an ammoniacal smell to the product. Also, there is formation of dimethylamine, DMA, and proteins undergo catabolism, resulting in the formation of ammonia, indole, hydrogen sulfide, and other products. Oxidation of the fish fat leads to rancidity, and this is not a desirable feature. So, let us see how the commonly fermented fish product like fish bagong is prepared. We will have a look of the preparation method. The first step is to mix salt with the fish or shrimp. Then it is placed inside a large earthen fermentation jar, and it is allowed to ferment for a period of 30 to 90 days, and during this period occasional stirring ensures that the salt is spread evenly or uniformly on the product, and then after the period of 3 months the product is ground into a fine fish paste which is ready for consumption. There are certain safety issues while consuming the fermented fish products. The most common issue is histamine poisoning, which is also known as scrombotoxicosis. It is intoxication primarily by Morganella or Proteus morganii, Klebsiella pneumoniae, Hafnia alvi, Proteus vulgaris, Proteus mirabilis, Enterobacter, Pseudomonas cepacia and Clostridium poisoning. The second concern is the Clostridium poisoning. It is also very often found and it is because of a spore forming anaerobic bacterium Clostridium botulinum which produces a heat labile toxin. Sometimes mold infestation also causes the spoilage of the fermented fish products. Aspergillus and Penicillium species are often encountered with the dried, cured fish product during their storage. The affected products show visible growth as well as discoloration. Some of the strains of the mold produce certain harmful mycotoxins like aflatoxins in the dry food product. Aflatoxins, you will be surprised, are one of the greatest health concern and is produced by Aspergillus flavus and Aspergillus parasiticus. Apart from the microbial infestation and spoilage of the fish product which are fermented, there are also other concerns like insect and mite infestations. The chances of insect and mite infestation are extremely high during the early stages of curing process. There are a number of parameters which govern the extent of damage by the insect and mites, and these are the temperature, moisture content, ambient relative humidity, the salt level in the product which govern the level of infestation by the flies and insects. The fermented fish products are extremely high with respect to the nutrient content. The fermented fish products such as sauces and pastes contain amino acids and polypeptides equivalent to about 10% protein. Occurrence of free unaltered amino acids contribute towards nutrition just like fish protein. These are also good source of calcium, iron and vitamins, especially the B group of vitamins. However, the safer use of fermented fish product is advocated because of their high salt content, and therefore the bulk consumption should be avoided and it should be used mainly as a condiment. In order to ensure the safety of the fermented fish product and taking into consideration the health concerns of the consumer, certain quality and standards for the fermented fish products have been advocated. The traditional preservatives such as salt, vinegar, smoke is permitted, but the use of antibiotics is not encouraged. Permitted coloring agents are also not allowed in the raw fish, but they may be allowed in certain unprocessed products. Use of polyphosphates is regulated by many countries and their maximum limits of usage have been set. Use of permitted antioxidants in fatty fish is allowed. So, students, let us sum up. Fermentation is a means to preserve the nutritional value of the fish, and also it is a means to enhance the flavor and its shelf life. Fermentation is an outcome of the activities of a number of microorganisms. These microorganisms are either working collectively or they act in a sequential manner. Their metabolic activities results in the production of active antimicrobial compounds such as lactic acid, hydrogen peroxide, bacteriocin, etc. It can also produce certain compounds like antimicrobial niacin, which is an effective means of preservation. Use of hygienic conditions during the fermentation process results in a product which is high in quality, which have been set up by the standards by international regulatory bodies. Thank you.